So next set of slides is about being precise. And it's about something that is gonna be relevant also for EMBL people, because we're gonna mention ORCIDs. So one key concept of metadata is that to be able to allow metadata to communicate between different projects, then they should be interoperable. This means that they should somehow be formal, accessible, shared, and use a broadly applicable terms and languages. For example, if I mention a specific piece of equipment, I should be specific enough for people to understand exactly which type of equipment I'm using, right? So this translates into actual IDs of things, if I do have any. If not, enough data to understand exactly what I'm speaking about or what I'm speaking about. And here in the slides, there's the example of an author name. So the name of a person is never, never, I would argue, specific enough, because it can be written in so many ways. For example, a name that contains also, letters that are not that do not belong to the Roman alphabet, then to the Latin one, then it can be written in even more ways. But any name, when I abbreviate it with just the first letter of the name and so on, so a name is never an ID. That is the main point. So ORCID. ORCID stands for Open Research and Contributor ID, and it's a free, unique, and persistent author-identified record that you can control. Maybe I can just try to understand here in the room how many of us have an ORCID profile or ID. Can we just maybe share this with the reactions? So can you use your like put your hand up if you do have an ORCID ID? Okay. Okay, I would say most of us, if not all. I can actually see you all, but I can check the participants. Oh no, not most of us. Okay, so if that's the case, I definitely suggest you to take a couple of minutes right now or after the course to create an ORCID profile. This is also linked to a whole initiative of OSiM. I don't know, Victoria, this is a bit of a unplanned thing, but do you want to have like share a word about it?
Yeah, sure. So EMBL requires that all publishing staff have an ORCID and keep it updated, so that you should not only have an ORCID, but also regularly claim your publications there, because it's the most clean and easy way to have all of your research outputs in one place. You can even claim things like preprints, your data sets, things like that. You can add your list of affiliations. And I think for an academic, having all of your publications that live there, it's better to have this as your record and your public facing CV than your LinkedIn, for example. Really recommend that everyone have it. And our goal is that everyone at EMBL would have an ORCID, and also keep it updated. If you have questions about how to claim your publications, you can do it through your PMC or just write to me. I'm happy to give you some tips and instructions for how to do that. But yeah, so it really is very easy, and I regularly update it. Whenever I publish something, I make sure to add it to my ORCID as soon as possible. And people do look at your ORCID profiles. So yeah, that's all I have to say about the ORCID side from EMBL's perspective.
So in addition to EMBL perspective, I would say that this is often the case. There are several institutions now that if they don't require, at least they monitor usage of ORCID, and this is becoming a thing. So if you don't have an ORCID profile, we highly suggest you to create one. So the exercise was about checking one of the papers of the lesson developers and checking how this is linked to their profile, and in their profile all their publications are linked. But I think since most of you know ORCID already, we can skip this exercise. You can have a look at it later on. So similarly to ORCID, other registers can be used to identify many biological concepts and entities. I'm sure you are at least familiar with one of them or more: the NCBI taxonomy, CABI for chemicals, UniProt, GenBank, and metabolic reaction and enzymes. So in the next couple of slides, we're going to have a look at organized taxonomies and in general how we can structure IDs. But first, we're going to go back to our table and identify all types of public IDs in the table. So this is exercise 2. It's already linked in the pad. And we can probably go back to breakout rooms to do this. Did anyone find a reference for the strain ID? Okay, so if not, I guess, no, we cannot consider the strain ID a public ID. It was an ID probably inside the experiment, but it's not a public ID linked to a control vocabulary online. What else? Not a unique convention for all funders, but there are funders that have grants IDs or even framework IDs. So they have IDs for their own, categories of funding, and then IDs for each specific grant that they found. And that's often the case that, for example, in publications, in the acknowledgement or in the funding section, these IDs are reported. But I'm not aware of a unique standard for all funders. I don't think that's the thing. Yeah. For example, the European Commission has IDs for all their grants, right? So you have a specific ID for the grant that you're also supposed to report every time a grant is involved somehow in a study. What else? I spot at least one other type of ID there, which is the DOI of both the protocols and eventually publications, right? That's also an ID. Anything else? Am I missing something? Genotype. Bioprot ID is basically the DOI, yes, and NProt ID same. And I guess we covered everything here, right? Type of thing in your experiment, you're probably referring to a control vocabulary. And control vocabularies can be of different types. So control vocabulary, the definition is that it's an enclosed and prescribed list of terms. The relationship between terms are not usually defined in the general control vocabulary, as terms are not. And usually the simplest form of a control vocabulary is just a list. It can appear in the form of a drop-down list, for example. This means that a control vocabulary, the general type of control vocabulary, doesn't really describe a structure for its own data. That's not the case. A little bit more or a little like structured are taxonomies instead. Taxonomy are control vocabulary, but arranged in a hierarchy. So terms are not usually defined, but the relationship between terms are usually defined. And the terms are arranged in a hierarchy. So for example, different species of bacteria, they these are terms that belong to the larger category of bacteria, so this is a taxonomy. Another type of control vocabulary, more specific, is an ontology. An ontology is a formal conceptualization of a specified domain. So terms are defined. The relationship between terms is also defined, allowing logical inference and sophisticated data queries. And terms are arranged in a hierarchy. An example is, it looks the same. So these terms are both arranged in a hierarchy and defined somehow. So well, I would now like ask you, does any example of any of these come to mind? Do you use any control vocabulary, any taxonomy, or any ontology besides the species names, because this was our example?
So when we do, for example, like sequencing annotation or something, we use gene ontologies. So like functional descriptors that are, I think, organized in the way an ontology is.
Yeah, gene ontology is an ontology because it also describes relationship between terms. Even if I would claim that most people use it as a control vocabulary, like they often, you don't even look at the structure of the gene ontology in like the tree of the gene ontology, right? It just mentions specific genes. Yeah. Okay. Any other examples? If not, I would say we can go to the next part.